Our next presenter is Manish Sadarangani. Manish Sadarangani is an international expert in translational vaccinology whose work uniquely encompasses laboratory, clinical, and epidemiologic research. His scientific expertise enables him to respond quickly to changing priorities, to tackle diverse knowledge gaps rapidly, and produce high-quality research findings to inform policy. Specifically, his research improves understanding of immune responses to vaccination, identifies high-risk groups to target immunization programs, and evaluates vaccine effectiveness to inform evidence-based policy. Welcome to the stage.
Thanks, Paige. So I'm really excited to be able to share some of the work that we're doing with you today. And for today, I really wanted to focus on the work we're doing using vaccines to prevent antimicrobial resistance. So I just want to acknowledge that I live here and work and play on the traditional, ancestral, and unceded territory of the Coast Salish peoples, including the Musqueam, Squamish, and Tsleil-Waututh nations, and I feel very privileged to be able to bring up my family in such a beautiful environment. So why do I care about antimicrobial resistance, which I'll refer to as AMR? So I'm a pediatric infectious disease physician. I see children in the hospital here in Vancouver who get admitted with serious infections. We send samples to the lab. We get reports back saying we found the bacteria, and then we get reports of which antibiotics we can use to treat them. And this is not one of ours, but this is the kind of thing that is becoming increasingly common, where we get a list of all the antibiotics that have been tested, and it's all resistant. So we can't use any of these antibiotics here. And so we're left with using antibiotics which are much less commonly, may not be available, and have much more side effects. And almost 10 years ago, the World Health Organization stated that the world is heading to a post-antibiotic era in which common infections will once again kill. Why I think you should care. So in 2018 in Canada, around a quarter of all infections were due to organisms that are highly antibiotic resistant. And with the current trajectory, that's estimated to go up to 40% by 2050. And as a result of this, the cumulative cost is estimated to be around 400,000 lives in Canada, and with about 120 billion hospital costs and almost 400 billion of lost GDP. On a global scale, this is a report published by the WHO a couple of weeks ago that I was involved in. So in 2019, around 5 million lives were lost globally due to AMR. By 2050, that's estimated to be up at 10 million with $100 trillion per year in terms of cost. Vaccines even now, could avert around 500,000 of these deaths each year. And we need a One Health approach. So I work in the box here and sort of the human health side of things, and there's others, as you've already heard a little bit about from this session, who are much better placed than me to comment on how we can intervene in agriculture, in the environment, in aquaculture. And so just to provide a bit of context, and on the right-hand side, this shows the link between climate change and AMR. And we know, and there's data that shows that as you increase temperatures around the world, we find increasing rates of antibiotic resistance. So the work that we do here at the Vaccine Evaluation Center, so I describe it as going from molecule to population. So I do research on a number of different vaccines, and each vaccine has a sort of natural life cycle, and depending on the vaccine and the disease, we do research in one or more of these domains, from population evaluation, surveillance, discovery science in the lab, to do preclinical studies and vaccine development, clinical trials of testing vaccines in humans, understanding immune responses, and then implementation and policy and vaccine hesitancy. So yeah, for COVID, we went around this entire cycle extremely rapidly over the last few years. But I'm going to talk about, for each of these domains, a bit of the work that we're doing related to vaccines against antimicrobial resistance. And to do that, I'm going to talk about these two bacteria specifically, which is a major focus for our group right now. So one is Klebsiella. So we don't currently have any vaccines available for humans for Klebsiella. And the other is Streptococcus pneumoniae, or pneumococcus, for which there are vaccines available and have been used in Canada, but have limitations. And for those who are not familiar, so these are bacteria that cause sepsis, meningitis, so infection of the brain, lung infections, urinary tract infections, quite disseminated infections, and often end up with people being in hospital, severely unwell, and sometimes fatally. So on the sort of surveillance side, there's some work that we've done here at the Children's Hospital showing that with any infection where there's significant antimicrobial resistance, the risk of death is doubled. So going from around 8.5% to 17%. And within these, there was a high burden of Klebsiella infections, which is really one of the reasons why we're focusing on this pathogen. This work is about to be expanded to the 16 pediatric hospitals across Canada through the Popcorn Network, and so we'll have national data to be able to inform what we do going forward. On the discovery side, we've been analyzing bacterial genomes from both of these organisms. We've had data on about 1,000 bacteria from both of these, and we've used these to try and identify what we think would be good vaccine candidates going forward. For Klebsiella, we've produced some prototype vaccines in the lab based on a nanoparticle technology that we have developed. And in mouse models, we've shown that the vaccines we've developed have given good antibody responses. And we're now moving this forward to see if they protect and expanding the testing through a newly funded consortium called Avenger, which includes looking at a number of pathogens and different vaccine technologies. In terms of the clinical trials, as I mentioned, there's no current vaccines available for humans for Klebsiella, but for pneumococcus, we've been looking at, in clinical trials, at the current vaccines to try and optimize the way they're used. So while they're not perfect, at least we should use them as best as possible. And then we're trying to understand better the immune response to these infections. So one of the underlying concepts of vaccine immunology is that if you can replicate natural immune responses, that would be a good thing to be able to do with a vaccine. It doesn't work for all pathogens, but for most it does. And so one of the studies that we're currently doing is called CLEAR, where we're taking samples from children who've had Klebsiella infections to look at what their immune response looks like and to see if we can replicate that in a vaccine. And finally, on the implementation side, as I mentioned, we do have pneumococcal vaccines that have been used. There's around 100 different types of pneumococcus, and the current vaccines can prevent a small number of those that were chosen because they were the most common. And in the blue and the orange bars, you can see that they have been largely successful in getting rid of the types that are in the vaccine, but they have just been replaced by other types. And so we're sort of constantly chasing our tail, and so the work we're doing is trying to design vaccines that will be able to protect against all of the different types. So the questions that we're going to address, and Paige is hovering, this is my last slide, is the burden of these infections in children and adults across Canada, so we can understand how bad the problem is and understand what the impact of vaccines is once we have them. Who are the populations that we should be targeting? So who are the high-risk groups? And in terms of vaccine development, the ultimate goal of this Avenger consortium is to have a new vaccine candidate ready for clinical testing within 100 days of a new pathogen being identified, and then hopefully to be able to test that in phase 1 clinical trials and beyond. So I just want to end by thanking my amazing team and all of the individuals and groups that have contributed to our research, and thank you for your time today.